Yeah,
हाँ वही
Rajendra said
कनेक्ट कर रही हूँ, कनेक्ट नहीं हो
the-
रहा लेकिन
that this is the best thing uh, that has happened to all of us. So I agree with him. Um, so my name is Murali. I used to be popularly called Chotu. We all used to have nicknames in those days, and I was the shortest of my batch, so I got nicknamed as Chotu. So some of you might have heard the name. So I'm currently an associate professor at Delaware State University, and we also have started the Delaware Center for Neuroscience Research. Uh, so to talk about my past CCMB life, I joined CCMB in 1990, and then finished in and left CCMB and India in 1997. I worked with Ashok Kar, and I worked on cloning and characterization of a tumor-specific antigen from the tumor AK5. I think every one of our students has must have used AK5. So, but during this time, I developed an interest, mainly because of the interest of and because of influence of my seniors, Sridhar and Rakesh, and a bunch of other people, Rajendra, Girish, Raghavendra. These names are must. I mean, these names are quite famous in CCMB. So I got an interest. I decided to take up a postdoc in neuroscience, which Ashok said that, hey, what are you trying to do? This is going to be like doing another PhD. And indeed it was. It was like had to learn a completely new system, completely new discipline, but it was fun. So that's the main part. That's what we learned in CCMB. If the CCMB culture still exists, and I believe, looking at you all, I believe it does exist. It's we do science for fun, and because it gives us a lot of pleasure. So Rajendra wanted me to give you all a quick introduction to where we are, where I am, and I live in Delaware. Delaware is a small mid-Atlantic state, and it's quite small because from north to south it's only about 160 kilometers. It has population of less than a million. In 2020 December, the census stated 930,000 people. So it's uh, even though its population is less, it's quite thickly populated. Dover is the capital, although Wilmington is the largest city, and Delaware has the best beaches on the eastern seaboard. Even Rajendra might not agree, but it has the best beaches. You should, if you ever come to the East Coast, Delaware is not too far. We're about 45 minutes from Philadelphia, a couple of hours from New York City, and a couple of hours from Washington, D.C. So it's easy access. And we are planning our next CCAB alumni meet, if that's going to happen when COVID is done, in Delaware and Maryland area. It has low cost of living. There's no sales tax. We have no airport or football team, but has an Amtrak station, so it's easy access by train. If you come to Boston or New York, you can take the Amtrak and then come to Wilmington. Uh, as I said, it's a small state. It's not very well funded by NIH. So two major universities. The University of Delaware is the bigger university. It's a private university, and Delaware State University, where I am located, where I work, it's a historically black college. It's funded by the state of Delaware. Historically black college or 1890s institution. At the end, after I finish my talk, I can tell you all if you're interested what it is. It's mainly has been established for black students who were denied admission into many southern universities. Congress decided to um, actually to institute a law. It's called the PL 480 law. Actually, there are few universities in India that were established under the PL 480 law. So this law extended internationally. So my master's was done from many agricultural universities were established under the PL 480 law. But that's history, and this is a neuroscience lecture, so I'll not digress. Neuroscience in Delaware got a big boost in 2010 when my senior colleague and my mentor, Melissa Harrington, who's a Stanford trained neuroscientist, uh, uh, obtained a NIH COBRI grant. A COBRI is Center of Biomedical Research Excellence. So this grant system was established to fund states that do not have a lot of NIH funding. So this grant was given joint, even though DSU, we are the lead institution, it was given to both University of Delaware and Delaware State University. and it funded about a dozen neuroscience researchers at DSU and at UD, and I was one of the 5 people who was recruited to DSU. So after I finished my PhD in '97, I headed to Boston. I came to Boston as a postdoc at Tufts Medical School, where I worked for about 7 years as a postdoc, and then decided to move to a lesser place, a smaller place, so that I could get a job, because Boston is extremely competitive. Those of you who are living in Boston, you know that it's extremely competitive. Everybody has a nature science, multiple nature science papers. So I moved to a smaller place, to Delaware, to University of Delaware as a postdoc, and then started establishing myself there. I worked there for 3 years before I got my first faculty position in New Jersey. And then, but 3 years later, I came back to Delaware because my family was still there. So I came to DSU. So if you have any questions, you can feel free to interrupt me as I give my talk. You can see that this is Lewis Beach, where I'm going to go today in the afternoon. So this is the famous Lewis lighthouse and beach. So I'm going on a boat trip today. It's my wife's birthday, so we're all going there. Right. So so without further ado, let me start. So actually, this project that how can I close this?
Click
Escape.
on your screen, it might go off.
Or escape on the left on the top. Escape button on the top left.
It's okay. It's fine. This is tripartite synapse. So I'm going to give a quick brief introduction to neuroscience. I'm sure all of you know this, but this schematic or cartoon shows a typical synapse where the axon and the synapse and the axon terminal that ends the presynaptic terminal that ends in the synaptic neuron. An action potential comes along the axon and then causes voltage-gated calcium channels to open on the presynaptic terminal, which lets in calcium, causes the synaptic vesicles, which contain neurotransmitter, to fuse and then release neurotransmitter into the synaptic cleft, which diffuses across and then binds to receptors on the postsynaptic membrane. And this can induce either an excitatory or EPSP or an inhibitory or an IPSP postsynaptic potential, which will further cause this postsynaptic neuron to fire an action potential. So this is common knowledge. This is textbook knowledge. But over the last 20 years, this idea has changed. The fact that there are glial cells, particularly synaptic astrocytes, that are present in the synapse, and then that they modulate neurotransmission or synaptic transmission, has been steadily accepted. The evidence for this is right now overwhelming. Now, astrocytes have been shown to be necessary for cognitive function. If you manipulate astrocytes, you can manipulate cognitive function in mice. So that's been, so how do they do it? We still don't know, but what we know is that astrocytes are not electrically active. They do not have voltage-gated sodium channels or potassium channels, so their membranes are electrically inert. So how do they function? Well, there are metabotropic receptors, that is GPCRs mostly, on the astrocyte membrane, which will respond to the neurotransmitter that's released. In this case, the major neurotransmitter in the brain is glutamate. So they can bind to glutamate via metabotropic glutamate receptors and can initiate a calcium flux within the astrocyte. And then this calcium flux can further cause these astrocytes to release their own transmitter called gliotransmitters. We call them gliotransmitters. And many have been identified. The most famous of them are glutamate, ATP, and many peptide neurotransmitters. So it's still, people are still trying to work out what's happening. So, and I'm one of them. So this, I wrote this commentary along with Michelle Jacob when I was a postdoc at Tufts, when Ben Barres, who is no more, had published his famous paper showing that astrocytes are necessary for neurotransmission. This was a science paper in 1999, from 98 to 99. So, or maybe in 2001. First paper was in 97, 98, but the second one was in 2001. Anyway, so this is the idea. So the idea that astrocytes can influence and modulate neurotransmission has existed, and evidence is still accumulating. Now, we wanted to test this idea. So how do we do this? So before I go into that, I'd like to talk about neural network activity. So what happens is when, as the brain develops, I consider myself a developmental biologist and a developmental neurobiologist. As the brain develops, the neurons are born in the subventricular zone. Uh, the nervous system develops from the ectoderm, from the embryo, and the subventricular zone first produces these progenitor cells, which give rise to both glia and neurons. First glia and then neurons, and then these neurons migrate to the 6 layers in the brain, and then they start networking. And once they network, they start network activity. So this network activity begins quite early, and depending upon the species, right? In chickens, its form, the network activity is seen by embryonic days 8 to 9. So this is spontaneous neural activity. Initially, this activity is randomized, and then it becomes highly synchronized. I'll show you what random and synchronized mean. Random activity looks something like this. So these are 4 electrodes from an extracellular recording multi-electrode array that we use in my lab. And you can see that these are the spikes. These are field electrodes. These are not electrodes that are inserted into a neuron, but they're placed outside. For when I talk to undergraduates, I explain this by saying that imagine a group of people in a large room having a party, and they're all talking to each other, and you have microphones on the ceiling that are, that you catch the sounds, and you would hear noise. That's what random activity would look like, and happens mostly in the awake brain. And that's what's happening. The people here are exchanging information. What's your name? What do you do? Where do you live? Kind of information. So this happens mostly in the awake brain, and the brain is processing sensory input and output. Whereas synchronous activity is highly synchronized. The spikes are highly synchronized, and it's like all of the neurons have stopped processing information and started singing. So sometimes these are called synchronous oscillations. And this is usually seen during sleep and anesthesia. Rajaram recently pointed out a paper by Jeremy Brown and Earl Miller, with whom Chanchal had worked. And they had, I met Jeremy Brown when he came to Delaware State University, and he talked about his brain activity research during anesthesia. He works at Mass General Hospital. So what we think is that this kind of synchronous activity that happens in the brain when there is no sensory input being processed, that is during sleep and anesthesia, is necessary for the functional organization. The brain is modular. You know that the occipital lobe has visual function, the parietal lobe has sensory motor function, the temporal lobe has auditory function. So we think that neurons that are functionally connected or will fire at a certain frequency, and they're synchronized. So these are being seen in what are called as thalamocortical oscillations during sleep. So I won't go into all of those details, but the mechanistic basis of how do neurons go from random to synchronous activity, that has not known, and that's very important for us to understand a neurological disorder called epilepsy. I won't go into details of epilepsy for lack of time. So epilepsy is a serious disorder that has no cure, there is no treatment, and that's because currently in the field, this is the current model, even though this is rather old, it's up in 89, but this is the current model that many epilepsy researchers actually still adhere to. So what they say is that there are recurrent connections between excitatory neurons, which are shown here by this triangle, and inhibitory neurons shown by this closed circle. And these recurrent connections cause epilepsy. How? There is a steady leakage of excitatory current via NMDA channels. You can't see the NMDA channel here, but this is the NMDA channel represented by this bar, and a steady leakage of excitatory current, that is via glutamate, into this into the excitatory neuron will cause this neuron to fire. And when it fires, it releases glutamate, or hopefully, and will cause the inhibitory neuron to fire and release GABA, which is an inhibitory neurotransmitter, and will shut this down immediately. So this process continues over time, And then there are other neurons that are connected into this network. And then they say the old adage, the old Hebbian adage, that neurons that fire together wire together. So this activity causes neurons to synchronize. That's the current idea. However, as a developmental biologist, I know that this is not how the brain looks like. There are astrocytes. So this is my best. I'm not an artist, so this is my best ability. I drew this myself of representation of astrocytes, and astrocytes are present everywhere, and they are there before the neurons are even born in the subventricular zone. They migrate first. They help neurons to migrate, and then they help axon pathfinding and synapse formation. And then now we know that they influence neurotransmission. So our idea was that if you could get rid of astrocytes and then have a purely neuronal network, what would happen? So that's the idea. And in order to do that, you need a system where you can grow neurons very easily. And when I came to Tufts, I got introduced to the chicken embryo. Chicken embryo has been around for 100 years now, more than 100 years. And it's a very beautiful developmental model system. I can talk about that later. But when I first joined Delaware State University, this was a side project that I wanted to do. And Carla, who joined my lab at the time, uh, started this work. And so basically what we do is, this is the forebrain, this is the optic tectum. So this is the entire chicken brain. We cut it out, and then we can dissociate the neurons using collagen or trypsin, collagenase or trypsin, and then we can plate them on in cultures and then grow these neurons. The best thing about a chicken embryo is they're much more easier, they're much more robust. Chicken optic tectum and forebrain neurons are very robust, you can grow them easily. So we wanted to grow these cultures and get rid of astrocytes. How did we do that? So this is a culture, a picture. It's a Nomarsky optics, or DIC optics as it's called, differential interference contrast. So you see neurons, bunches of neurons with their axons, and there are astrocytes which are much flatter cells. You can't see them. So this is a normal chick embryo E7 embryonic day 7 culture. So we tried many methods, but we finally chose to use a 5-fluoro-deoxyuridine, which is a mitotic inhibitor. Neurons are post-mitotic, whereas astrocytes continue to divide throughout their lifetime. So FUDR would kill off astrocytes, or glial cells mostly, and then would spare the neurons. However, neurons do not survive for long in the absence of astrocytes because they depend on them for trophic factors. So you can maintain them like this for about a week, but we wanted to maintain them longer to look at long-term network activity. And in order to do that, we decided to supplement them. We took a lesson from Ben Barris's old paper, where he supplements conditioned medium, the medium with conditioned medium. We normally grow neurons in neurobasal medium, and when you add FUDR, astrocytes die and neurons kind of start dying off. But when you add astrocyte conditioned medium to this, neurons are fine. We can maintain them. I maintain pure neurons for 6 months or longer in culture, and they grow fine. And so we stuck with this idea. However, as reductionists, as molecular biologists, we know that we can only change one thing at a time. So we didn't want to. We had to add ACM to control cultures also. So these are the two conditions that I'm going to talk about that we've used. Although we checked that this condition with ACM and without ACM, without FUDR, they're very similar. Their network activity patterns do not change. So we wanted to confirm that these FUDR treated cultures indeed have neuronal networks. There's no difference whether between the neuronal network. If you see, neurons are here in red, we label them with beta-3 tubulin, and GFAP is a marker for astrocytes, a cytoskeletal protein. So control cultures without ACM, normal cultures, and added ACM, there's not much difference among them. And when you treat them with FUDR, astrocytes die, but neurons are quite healthy. How do we know that neurons are healthy? Is we did a bit of electrophysiology, a whole cell patch clamp. Delaware State University has a electrophysiology core, and which is situated right across from my lab. I helped establish this, so it's easy for me to do it. So we find that the resting membrane potentials and action potentials that are generated, the spike amplitudes, and ability to generate electrode action potentials using a voltage clamp, they're all identical between control and FUDR treated cultures, meaning that these neurons, when you remove astrocytes, individual neurons are healthy, right? They can fire action potentials, and as long as we supplement them with astrocyte condition media. So we wanted to use these conditions. So we use these conditions, and then we grew these cultures on multi-electrode arrays. So there are many different companies now that sell them, that make these systems. So what it does is it has a small culture dish with electrodes. These are titanium black electrodes from this first company that started this Med64, and you can grow these cultures. And these extracellular electrodes would record activity. So the system that we had used at the time contains 64 electrodes in an 8x8 grid right here, and then we can grow our cultures. So you have without FUDR, which are the control cultures, and with FUDR, which are treated cultures. These are pure neurons, and these are neurons in the presence of astrocytes. So with this, how does this electrode activity look like? These are control cultures at 14 days in vitro on Med64 plates, and each of these channels represents one electrode. And I'm going to press enter. I hope you can see the spikes. You see that this activity is mostly random, seems mostly random here, right? However, by 21 days in vitro, these are control cultures again. These cultures now are synchronized. We call this synchronized bursting. So any network of neurons from invertebrates to human cultures will synchronize. With left alone in culture, they will form networks because obviously they do not have any sensory input. They fire, and then they become synchronized. Whereas FUDR treated cultures at 21 days in vitro, these do not synchronize. They're still random. So this was the first piece of data that Carla generated. And then we have been able to repeat this multiple times. And we also did an experiment where we can add astrocytes back into this network, which is actually not very easy to do, because when new astrocytes are added, the network breaks up, all the neurons start floating up, they die. But we could control the number of astrocytes, and then we were able to repeat this experiment a couple of times. And when you add astrocytes, some synchrony comes back. So here's what basically this is the data. So control cultures in the presence of ACM, they will synchronize by 21 days in vitro. These are 4 representative electrodes that we're showing here, whereas FUDR treated cultures will never synchronize. We can keep them as long as we want, a month or more, and they would not synchronize. 2 months. I said that I had maintained this culture for 6 months. It's not because these FUDR treated cultures are firing less, or the neurons are not firing action potentials. The weighted mean firing rate, which gives us an estimate of the network activity, is generally similar or slightly more than control cultures. So these neurons, in the absence of astrocytes, are firing action potentials, and their activity is actually slightly more. So the only difference is that they're not able to synchronize. So we have a measure, we developed a measure, well, not in my lab, but in my collaborators' lab, what we call a Synchrony Index, that will give us a measure of synchrony. So anything above 50% or 0.5 Synchrony Index is considered synchronous. I can go into, for those of you who are interested, I can talk a little bit about how we do this, although I'm not an expert. But we find that FUDR treated cultures have a very little synchrony. They're firing action potentials, but they're not synchronized. This is another experiment that we did to show that control cultures without ACM are not very different from control cultures in the presence of ACM. Essentially, these 2 experiments were done separately, so we show this data separately. So what we've shown so far is that if you get rid of astrocytes and keep neurons alive with conditioned medium, they will fire action potentials. They are healthy, but they will not synchronize. So if you think of synchrony as a neurons as a bunch of musicians that are playing music, and then you need a conductor, and that's why I named this title Astrocytes, or the conductors of the neuronal symphony. So one of the questions that we asked was, is there a critical window period during which astrocytes are needed to train the neurons to synchronize, and after that they don't have a role? Remember, this is all in vitro, right? We are trying to find a mechanism in vitro. What happens in the brain is could be is much more complex. So ideally we would have wanted to add astrocytes at different time points to these pure neuron cultures to see if synchrony would develop. But that was a difficult experiment and we couldn't do it. So we did the converse. That is, we added FUDR at different time points and then measured synchrony at 21 days or 3 weeks or 4 weeks. And then we see that synchrony index at whatever time you are. Even though these cultures were synchronous before, when you add FUDR at 14 days, they lose synchrony. And the weighted mean firing rates are not very different. So they're still firing action potentials. So what's happening here? So it's something like if the conductor has started conducting the music and then he goes out for a smoke, the musicians will lose their sync. Right? So astrocytes are needed at all times, not only to generate synchrony, but to maintain it. So that's what we think. So this data right now is it's under second revision. We're fighting to get it published in Journal of Neuroscience Research. So hopefully the next time we meet, I will be able to share the paper. So to summarize, in the absence of astrocytes supplemented with astrocyte conditioned medium, neurons are healthy, but they fail to synchronize. And astrocytes are needed for the generation of synchrony as well as its maintenance. So how do they do it? So current research in my lab is we are focusing on that. We're focusing on GPCR and the GPCR mediated calcium elevation in astrocytes. So this idea that GPCRs will cause are necessary for astrocytes to respond to neurotransmitter and then to release their own neurotransmitter has been around. There's evidence from multiple systems, but whether this is the way that astrocytes can actually cause neural networks to synchronize, that is the bigger question. And that question is important because then we have a new target to treat epilepsy. The cause is in the astrocytes, not in the neuronal network. That's what we believe. So how do we do this? What are we doing right now in the lab? We are using a dominant negative strategy. I remember I'm trained as a molecular biologist from CCMB, so this would be my first. And normal, there's a lot of evidence in the field using pharmacology, using drugs, but pharmacology is rather dirty. And a lot of my neuroscience colleagues will get mad if I say this out loud. But pharmacology is dirty. We do not know what ectopic receptors the drug is acting on, whereas a molecular approach is very precise. That's what PMB taught us. Right? So we are now right now focusing on metabotropic glutamate receptors. There are also the purinergic P2Y1 receptors, the ionotropic ones, the channels are P2X receptors. The GABA A receptors are ionotropic, but the GABA B are metabotropic. They're all GPCRs, and then so our idea is to co-culture, you make dominant negatives to block these pathways, and then co-culture the neurons with MEAs with the dominant negative astrocytes. We came up with a method of co-culturing them, and I'll tell you about this in the end in a couple of minutes. So this is the model that I developed. I drew this myself. So my artistic skills have been getting better. So what we think is happening.
Yeah, it's beautiful.
What we think is happening is that at a synapse, neurotransmitter is released at the tripartite synapse. Metabotropic glutamate receptors respond, and then they initiate a localized calcium flux in the astrocyte process. Astrocyte is a complex cell with multiple compartments. So we think that this happens in this flux happens here in the process. And then when it crosses a certain threshold, it leaks out and calcium goes into the center of the soma, so-called soma of the astrocyte, and will cause a calcium-induced calcium release from endoplasmic reticulum stores. And this calcium, there's a lot of calcium that's released. And I'll show you a little bit of evidence of that. And this calcium will now travel and then go to other astrocytes via astrocytic gap junctions. That is step 4. So this calcium-induced calcium release can also cause neurotransmitter to be released at multiple synaptic sites, shown here by 5, causing all these neurons to fire. So neurons that fire together, wire together. So that's how we think that astrocytes entrain neurons to fire synchronously. So how do we test this? So we've been generating, so interestingly, all these pathways use the common G alpha Q G protein. And this is a standard canonical pathway. And the only difference could be that certain types of astrocytes might be releasing ATP via a different pathway. So we don't know that yet. So right now we are working with Embryo dominant negatives. It's a truncated metabotropic receptor. We're also looking at blocking glutamate release by astrocytes. VAMP2 is synaptobrevin of the snare complex. This is the complex that is necessary for the fusion of the synaptic vesicle with the presynaptic membrane. And finally, we are using optogenetics. That's a new craze. So if I don't jump in on the bandwagon, I'll be left out. So we started using optogenetics, controlling neurons by light. That will be a different lecture at a different time point, I guess. How optogenetics has revolutionized neuroscience. What we can do is we can control neuronal firing using light activated channels. Melanopsin is a G protein coupled receptor that's been recently discovered, and we are using that. So what's happening here in the next couple of minutes? We measure calcium activity using GCAMs. These are genetically modified calcium sensors.
क्या बोल रहे हो? तुम्हारा 30 पूर्णिमा 26.
So GCAM is a genetically enhanced calcium sensor, a fusion of GFP with the calcium, the EF hands, which Balu's group who have been working in quite familiar with. So this is a calcium sensor. So what happens is that when we add glutamate, you can see that this culture of astrocytes follows. You can see calcium activity. Whereas when we express the embryo dominant negative, these cultures do not, or divide minimally. Just to ensure that these neurons are fine, we added ionomycin, which is a calcium ionophore. That we're not seeing, there's nothing wrong with these. So ionomycin induces, it's a calcium ionophore, which punches holes in astrocyte membranes, and then calcium comes in and it causes a calcium-induced calcium release. So we effectively show that embryo dominant negative blocks the embryo pathway, the embryo GPCR pathway. We also have data that shows that if you block VAMP2 or synaptobrevin, a glue sniffer is a similar glutamate sensor, GFP, with a bacterial glutamate transporter periplasmic protein. So whoever made this had a very good sense. Glue sniffer, very interesting protein. Here we see that this glue sniffer actually is situated extracellularly. So it's sitting outside the astrocytic membrane and is sensing glutamate that's released by astrocytes. So when we add ionomycin, you can see that there is a flash. And the dominant negative, excuse me, that's when ionomycin was added. Very little activity. So this work was done by Toby in my lab, a master's student, and he has repeated this experiment a few times, and then he hasn't given me the collated data yet, but this is from data from a single experiment. So we measure relative fluorescence, and we see that the dominant negative effectively blocks it. So Tanner Clifford, another graduate student in the lab, is developing the melanopsin astrocytes. So this is again we measure calcium activity with GCAM6F, and a picture on the right. I don't know why this was first, but this red circle here shows we actually give it a bleaching pulse of 470 nanometers light, and there's not much change in the fluorescence. Whereas these cultures to the left, the 3 or 4 astrocytes here, when the bleaching pulse is given, they induce, I love to watch this. So what we are seeing is we believe is a calcium-induced calcium release. So since this is a strong pulse, once a small amount of calcium comes in, this process continues to happen until all the calcium in the medium is depleted. So Tanner had shown, so we're able to induce a CICR with light, and we're able to block it. We're hoping that we'll be able to block it with one of the dominant in combination with any of these. So this data is still underway. We got this in 2020, and then COVID hit, and things in the lab slowed down. So, but now we are catching up. We're starting to come back into the lab and then do these experiments. So the next time we meet, the next time I get a chance to talk to you, great guys, I hope to give you more data. But the idea that what we're going to do is we're going to take these dominant negative astrocytes and then co-culture them with dissociated neurons. We cannot replace astrocytes in these, but we have shown earlier that if you add fully differentiated astrocytes, these will integrate into the network, and then they will be right there, and then they will be at least 50% or more of this network, and then they would cause a severe, they would prevent neurons from synchronizing. At least we expect not to see synchronous activity within these electrodes where these astrocytes are present. So this is our goal and this is our idea. So stay tuned for more data on this. I'm going to stop here. I think I've taken a few minutes more. My collaborators, Rhonda Zakpasu is at Georgetown University in Washington, DC, and she's a physicist and a computer scientist, and who also does research, epilepsy research in her lab. Melissa Harrington, who's my mentor, she's now currently Associate Vice President for Research at Delaware State University, and this project started in her lab. Denny Galileo, who is also a chicken embryo expert, I worked with him as a postdoc for 4 years. And Surya does our he's a brilliant statistician who does all our statistical analysis for us. So I don't have to, I just sit in the work. Ajay, the postdoc who joined my lab last year, he's the one who did all of these, who made these dominant negatives and did the calcium imaging, live calcium imaging. Carla just graduated. She's a PhD student. She's my first graduate student to graduate from my lab. Monet, Toby, Tanner. Monet just graduated and she's now at Georgia Tech. Toby and Tanner are master students in my lab, and Gabriel is an undergraduate who also graduated and left. We have an imaging center, and Michael Moore is the director, and he and Ajay work together, and they're all great guys. So I was supported by an initial NIH COVID grant of $50,000. This whole work started with that, and then was funded by a HBCU Research Initiation Award from NSF. And now I have a NSF grant, the Neural System Cluster grant, that's funding my work. We'll hopefully soon get an NIH grant. And this is Carla, and that's me at the back in the hooding ceremony, which happened a couple of weeks ago. So I'm going to stop here and take any questions that you may have. Thank you so much for your attention and for this opportunity to interact with all of you.
Yeah, it's a beautiful talk, Dr. Murali.